Hello everyone, my name is Dale Begley, and I am a scientific curator in the Mouse Models of Human Cancer database at the Jackson Laboratory. Today's video is going to be a tutorial on the cBioPortal for cancer genomics. The cBioPortal for cancer genomics provides visualization, analysis, and download of large-scale cancer genomic datasets. Mouse Models of Human Cancer database has instituted a specialized instance of the cBioPortal for large-scale PDX datasets. Patient-derived xenograft models, or PDX models, are generated through the engraftment and passaging of tissues or cells from a patient's tumor into an immunodeficient or humanized mouse host. PDX models are an important in vivo platform for cancer biology research and for preclinical testing of cancer drug efficacy. The Mouse Models of Human Cancer database cBioPortal PDX instance allows users to access to molecular profiles and clinical attributes from large-scale PDX cancer genomics datasets and user-defined subsets of these larger datasets. These data can be organized and analyzed in a variety of ways. The cBioPortal includes many data types, some of which are included are DNA copy number, mRNA microRNA expression or protein level and phosphorylation data, DNA methylation data, or de-identified clinical data. Now these depend on the data set. Some have all of these, some have only a few of these. The Mouse Models of Human Cancer database can be accessed from the Mouse Genome Informatics website by clicking on this bar here with the red arrow sign in, or you can go directly to the Mouse Models of Human Cancer database website at the highlighted URL up here, tumor.informatics.jax.org. Now, once you're there, you see the basic design of the website with a top Mouse Models of Human Cancer database. The American Cancer Society facts table here shows you the highest ranking incidence of different tumors. And we also have different pull-down menus that will give you different information. The Searches and Tools menu allows you to go and click, following the red arrow, click on the cBioPortal for PDX, which is where our instance will be found. Now, if you click on that link, you'll find here is the website for the cBioPortal instance. You'll see it has various structures in it. You'll see it has a list of the different data sets we have downloaded and the two different types of general types of queries you can do, all of which can be personalized, and it has all sorts of the number of samples in each of these data sets. If you click on the datasets tab up on the top, it will give you a little more detail on what each one of these datasets has in terms of number of samples, number of mutations, CNA data, etc. If you go back to the home page, you'll see the way to access the different data is by one, you need to select which data set you want to query, and you can select more than one or all of them. And you can either query by gene, where you would query for information from a specified gene in each of those data sets, or a general survey of the data in that data set. So we're going to do a query by gene first. And there you see the query by gene page. And what you need to do is need to type in a symbol. The symbol needs to be a valid human gene symbol, and it's validated automatically. So if you type in KRAS, and it doesn't, don't have to have it the proper case. They can put it in lowercase. It'll still figure it out that it's a proper gene symbol. And when this turns green, then proper symbol. Now you can, will search all the cases in the study. You can actually enter specific subsets of that data set, which we'll do later. If you hit query on a different video, you get back and return a result, which is all of the samples that have KRAS data in that particular data set. And this is shown to you in something called an OncoPrint view. The OncoPrint view shows you different tracks and different pieces of data. These tracks can be modified. If you click on this in live setting, the tracks you can add or delete different data sets. These hashtags, as it's written here, each grouping of hash marks is a different efficacy study. So this is one study, that's another study. Each column, up and down, represents treatment results for one of the models in the study. The color coding determines the response, like different cancer types here. Like this is an endosquamous carcinoma. That's actually lung carcinoma. That's a carcinoma. Also, treatment response. See the treatment response. That's a total stable disease, progressive disease, a no data, etc. Now, what we'll show an expansion that you can see here. If you hover over one of these hashtags, a box will open up that will tell you what the data for that hashtag is. So this one would be for the specific sample, TML1594, treated with Avastin, and the treatment response was a stable disease. Now you could, through the treatment description, study ID, the cancer type, they'll all have pop-up windows that give you data. Now, if you go back to the cBioPortal to try to do the Explore Selected Studies, you'll see the Jackson Lab. You would choose the Jackson Lab ID model set again, click on Explore Selected Studies, and you go to what we call a study summary page. This is a visual summary of all the data in that particular sample. Number of samples per patient, patient per sex, patient sex. These are treatment results for the patient. These are the patient age, all the demographics, the primary site, tumor site percentage, etc. And these can be customized. You can choose here, you can add or subtract different types of data. Now, if you click on the clinical data tab here in the upper left, you get to a page that lists all of the patients and or the samples and the patient IDs with them. And if you click on these individual connect links, you will end up getting more detailed information on each patient. So in future videos, we'll give you more details on how to do this. And in a second, we'll do a small live demonstration. Now, if you have questions, at the bottom of every page, Mouse Genome Informatics web pages, including all the pages on Mouse Models Human Cancer Database, you have a support link. So you can send questions and comments to MGI user support. Now, the Mouse Models Human Cancer Database staff consists of Carol Bolt, who is the PI, myself, Dale A. Begley, and Deborah Krupke, who are the curators, and Steve Newhauser, who's our software person who writes all the code. Now, what we're going to do now is do a small live demo. You can see that what I showed you. This is the MGI homepage. You go to the Mouse Models of Human Cancer link, and it takes you to Mouse Models of Human Cancer. And then you pull up the menus, and you click for the cBioPortal for PDX. And hopefully it'll open, and it does open. And you can see we've shown it before. If you click on that particular page, query by gene, put in KRAS, that's a valid symbol, search, you get query come back. And the part you need to change the view to be events per sample in order to have it to look right, or else it just views all the way across. Now this ends up showing a much more reasonable presentation. You can see you have the pop-up window for cancer types, glioblastoma, stable disease, and that's the treatment of the Avastin. These are all sorts of different treatment responses, and you can see the same tumor has different responses. Sometimes it does, sometimes it doesn't. And if you look at here, you see you can change what the track is. You can add final diagnosis, update the tracks. And see you added final diagnosis. We'll go into more detail on this later. We're just doing a general demo right now. Now, as we said before, you can also explore a selected study, which gives you the summary page, as shown right here. You can see samples per patient, and you have pop-up windows, which tells you more details in that in a summary fashion, the race, primary tumor sites, etc. And you can move these around if you want to change the presentation, and you can also add or delete different data from there. Now we'll present more detailed presentations on how to use the different functions of this at a later date. And I thank you very much for attending this seminar and watching this video, and hope you have a wonderful afternoon. And if you have any questions, please feel free to ask.